Hello, everyone. So, today is our sixth histology seminar on cardiovascular system. We'll be talking about the formed blood elements and then getting back to the cardiovascular system. So, I'll let Varun start off.
All right, guys, so we'll just begin with the simple description of blood. As you guys know, blood is made up of two components. We have plasma and the formed elements. So these formed elements, they're divided into different categories. We have erythrocytes, leukocytes, platelets, and then we have these leukocytes, which are divided into two more further categories. We have granulocytes and agranulocytes. And then granulocytes include neutrophils, eosinophils, basophils, and agranulocytes include monocytes and lymphocytes. This diagram is just from your lecture and it's just like, basic introduction. So now when we talk about plasma, what is plasma and what it is made up of? It is a clear yellowish liquid in the blood that acts as a medium for transporting nutrients, water, nutrients, waste, and other substances. So it's just like, a medium for other substances other than blood cells to travel. So it includes proteins such as albumin, globulins, fibrinogen, and it contains organic compounds as well as inorganic salts. When we talk about like the basic function of these proteins, we have albumins, which keep the fluid balance in the blood vessels. Then we have globulins, which are divided into alpha, beta, and gamma. And when we specifically talk about the function of gamma globulins, these help fight infections as they produce specific antibodies. Then we have fibrinogen, which is important for the clotting of the blood. And then the organic compounds include amino acids, vitamins, and hormones. Now, when we talk about the first type of blood cells, we have the erythrocytes. Most of you will already know this. They are a permanent component of the blood. They are primarily responsible for the oxygen transport, and they do not have a nucleus. Their shape is disc biconcave shape, and they have an indent in the middle, which increases their surface area to volume ratio in order to allow a faster gas exchange to take place. The main protein or the main component of the erythrocytes is hemoglobin, and it makes around 33% of the cell's content. And the different types of hemoglobin include adult hemoglobin and fetal hemoglobin. You learn about the difference between both of them later. So in this diagram, you can just see the basic characteristics of the red blood cell. You can see the disc biconcave shape. This represents the hemoglobin. And since they do not have the nucleus, this provides them a bigger space to allow more oxygen transport to take place. And then when we talk about conditions related to erythrocytes, we can have erythrocytosis or polycythemia, which is just the increase in the number of erythrocytes. And then when we talk about anemia, it's opposite. It's decrease in the number of erythrocytes because of reduced hemoglobin concentration. And this can be due to conditions like hyperchromic anemia as well as sickle cell anemia. Now, one form of erythrocytes is the reticulocytes. These are just nothing but immature erythrocytes, which within like 24 to 48 hours, they convert into erythrocytes. They contain ribosomal RNA and they gain energy through anaerobic respiration. When we talk about the lifespan of erythrocytes, their lifespan is 120 days, and after that, they're broken down by spleen macrophages, and again, all your erythrocytes are replaced again. When we talk about the cytoplasmic membrane structure of the erythrocytes, it contains proteins which are linked to the cell cytoskeleton, which helps maintain the shape of the erythrocytes and its flexibility to allow faster diffusion of substances to take place. Then, when we talk about leukocytes, these are just your body's defense systems, and it is important to remember that they are not permanent in your bloodstream. Their main function is defense against infection, so they only come into your bloodstream when there's an infection inside your body. So they move to the tissues through a process called diapedesis. You don't need to know much about this for now. Just know that this process allows them to move out of blood vessels into the connective tissue wherever there is damage. When we talk about the count, the number is 6,000 to 10,000 per microliter of the blood. Again, not important for you guys to remember now. And then when we talk about types of leukocytes, as I said before, we have granulocytes and agranulocytes. Granulocytes, they contain visible granules, and agranulocytes, they do not have visible granules. So when we talk about granulocytes, they have two specific types of granules. These categories are specific granules and azurophilic granules. So specific granules, they serve specific functions in immune response. And when we talk about azurophilic granules, they function as like primary lysosomes that phagocytose the bacteria and help break it down. As I said before, types of granulocytes are neutrophils, eosinophils, and basophils. So now they can be stained with different types of dyes. So basophils, they can be stained with basic dyes. It's in the name, BB, basic dyes for basophils. Eosinophils, they are stained with acidic dyes. And neutrophils, they do not attract both either acidic or basic dyes. When we talk about the shape of the nucleus of the granulocytes, it's a variable shape. That's why we call them polymorphonuclear leukocytes or polymorphs. Polymorphs mean that there's just the variation in the shape of the nucleus. And then this table is just the summary of the differences between neutrophils, eosinophils, and basophils. So as we talked about the granules, in neutrophils you have specific granules and eosinophilic granules which are involved in primary response. Then we have eosinophils, they have specific eosinophilic granules and another component called major basic protein. And then when we talk about basophils, they have specific basophilic granules as well as eosinophilic granules similar to the neutrophils. When we talk about the difference in the nucleus, neutrophils have a highly segmented nucleus. That's why they have 2 to 5 lobes. And when we talk about eosinophils, they are bilobed, 2 segments. And then when we talk about basophils, they are irregular shaped. As I said before, neutrophils they provide defense against microorganisms by the process of phagocytosis. Eosinophils, they're involved in the process of inflammation, and basophils, they respond to allergic or inflammatory responses. And when we talk about some specific characteristics of these cells, when we talk about neutrophils, they have bar bodies in females, and they show this band form when they are immature. When we talk about eosinophils, they contain enzymes which include acid phosphatase, peroxidase, as well as major basic protein, as I said before. And basophils, they contain heparin, histamine, and peroxidase. Now, when we talk about agranulocytes, so they cannot be stained by dyes. We have lymphocytes and monocytes. So the nucleus, they are mononuclear. Both of them are mononuclear, but the lymphocytes have a spherical shape, and monocytes they have oval or a kidney shape. Both of them basically have a pale basophilic cytoplasm with azerophilic granules, and no specific. It is important to remember that they have no specific granules present. When we talk about the function of them, lymphocytes, they're involved in specific immune response, which means that we have B cells, which are involved for humoral immunity, T cells, which are involved for cellular immunity. You'll learn about this for your next micro, so next week. And yeah, they are highly motile when activated and they can enter the bloodstream. When you talk about monocytes, they're part of your non-specific immune response. And they're involved in phagocytosing of the debris and the bacteria, and they act as antigen presenting cells for lymphocyte activation. So here is diagrammatic representation of all of them. One thing I forgot to mention is that lymphocytes, they are the smallest cells inside your body, and then we have the monocytes, which are the largest cells. And here you can see neutrophil has 2 to 5 lobe nucleus, hypersegmentation, and then we have eosinophils. They are bilobed. So this you can use this diagram as a reference to compare all of them. And lastly, when we talk about platelets, they are small disc-shaped fragments that originate from large bone marrow cells called megakaryocytes. So they're just mature platelets come from megakaryocytes. And important feature about them is just like erythrocytes, they do not have a nucleus and they have 2 different structural zones. One is the granulomere and the other is hilomere. Granulomere is the central part, which means this part or this part, which contains granules with clotting substances, and the hilomere is the transparent outer region here, which contains microtubules and actin cortex, which helps maintain the shape of the platelets and helps in clot retraction. Clot retraction would be like just removal of the clot after the damage has been healed. Then when we talk about the count of the platelets, it's around 150 to 300,000 per microliter. And the basic function would be that they play a crucial role in blood clotting by forming the platelet plug, releasing chemicals to strengthen the clot, and aiding in clot retraction and removal after healing. So that was it. And if you guys have any question, please.
Okay, so I'll be speaking about the circulatory system. And before I begin, I just want to say that whatever we'll be explaining right now is going to keep on coming up until the end of your summer semester, basically, because these are basic differences which you have to know, and professors will keep on asking you this in all the tests which you have. So, in the circulatory system, it is divided into two types. There is the macrovascular bed and the microvascular bed. So, as we know that macro means something like large, and micro is something really small. So, macrovascular bed by default then contains of these large arteries and veins, whereas the microvascular bed contains like small arterioles and venules. So the microvascular bed, it basically allows like the production and the circulation of all the tissue fluid, and this by default then enables the exchange of substances between the bloods and the tissues. This is divided into three types. So the first type is going to be the common type. So what this basically means is that the blood flow is going to be first through the arterioles, then it goes into the precapillaries, then to the capillaries, then the postcapillary venules, and the venules. So that is going to be your common type of the blood flow. Next is the portal systems. So basically, the key feature of the portal system is that there are two consecutive capillary networks which are like in two loops. So since there's two loops, one of them is always going to be of the common type, which I just explained, and the second one is either going to be arterial or venous. So the arterial portal system, it works only in the kidney, and the blood flows in like first into the arteriole capillary and again into the arteriole and then continues into the capillary and venule. So since this is arterial, capillary, and arteriole is going into the arteriole twice, so this part of this chain is going to be the arterial portal system. Now, arterial capillaries and venule, like I explained before in the common type, this looks like it's following the actual way it's supposed to follow the blood. So this is going to be the common type chain. Now, in the venous portal system, it works only in the livers and the adenohypophysis, and essentially is pretty much the same thing. So, arterial capillaries venule, since it's following the normal order, it is going to be the common type of the network. Whereas over here, venule capillaries venule, this two types, two is going into venule again after this capillary. So, this is going to be the venous portal system. The third type are the atriovenous anastomosis. So what happens here is that the arterioles and the venules is fused with each other and they completely omit the capillary network. So that's why it is called the atriovenous anastomosis. Now let's talk about the general description of your arteries and veins. The arteries are essentially of two types. There are your elastic arteries and your muscular arteries. The elastic arteries are pretty large, and for example, like your aorta, and they actually have a pretty thin wall. And moving on to the muscular arteries, they are usually medium and small sized, and they are like most of the arteries in our body except for like the really big ones. Vasa vasorum, these are these really small, thin-walled arteries, which their function is basically to supply the walls of these larger arteries and veins. When we talk about the veins, there is the muscular veins, which are pretty large. For example, we can see the pulmonary trunk. Fibromuscular veins, again, they're medium and small sized, and pretty much most of the veins in our body are fibromuscular veins. Now, the brain sinuses have got a different type of vein, which is called as a fibrous vein. And the important thing about it is that there is absolutely no muscle tissue present in any of the walls. So let's talk about the structure of an arterial wall in detail. So since muscular arteries are the most common type of artery, so this is what you will be able to see when you see a slide of a muscular artery. And essentially all the arteries and veins, they have the common three layers, which are the tunica intima, tunica media, and the tunica adventitia. So this innermost layer you can see here, that is going to be a tunica intima. This middle red thick layer you can see, that's going to be a tunica media. And this outer layer is your tunica adventitia. So I'll use this diagram so that you guys understand it better. The tunica intima is this part, which consists of the endothelium and the basal lamina. And of course, it has a subendothelial layer, which consists of a loose connective tissue, which is longitudinally arranged. And this line, this thick line, which you can see here, the specific one, that is called your internal elastic lamina. And this function is basically just to separate the tunica intima from the tunica media. Now, when we talk about the tunica media, it is circularly oriented. And it consists of, like, as you can see, it's like pretty thick layer, and it consists of smooth muscles and fenestrated elastic membranes. So the word fenestrated essentially means that the elastic membranes have got a bunch of holes in them. And these holes are pretty important because, firstly, they help make the artery more flexible so that the blood flow is very easy for it. And another function of it is that since there's holes, the vasa vasorum, which supply this artery, all the nutrients from it, such as the oxygen and like all the other nutrients from the blood, they can easily get absorbed into all of these smooth muscle cells because since it consists of smooth muscle, obviously all the muscles in our body need basic nutrients from the blood as well as oxygen, right? So it's just going to be easier for all of these nutrients to easily pass through them and enter even like the deepest layers of the smooth muscle. This part, you can't really see it clearly, but it is called as the external elastic lamina, and it is not really an important thing which you need to know because most of the times it's not even present on the slides. You can't even distinguish it. But again, its main function is just to like separate tunica media from the tunica adventitia. When we talk about the tunica adventitia, it's pretty basic. It's a pretty thin layer, and it consists of loose connective tissue, which is longitudinally arranged. One thing you need to know about these three layers is that the first layer, tunica intima, this part is always going to be longitudinally arranged, and tunica media is circularly arranged, and then tunica adventitia is again longitudinally arranged because this is like something they keep on asking you in all of your e-tests, like micro test, or for example, even like a final test. So this you need to know, like the orientations. Now, structure of the venous wall is pretty much similar to the structure of the arterial wall. And again, this part is the endothelium, and it also has a basal lamina. The subendothelial layer consists of longitudinally arranged loose connective tissue, and it also has an internal elastic lamina, which separates the tunica intima from the tunica media. Tunica media here, it has a circular orientation again, and it is very thin compared to an artery. It consists of collagen type 3 loose connective tissue and a few strips of smooth muscle here and there. When we talk about the tunica adventitia, it is quite thick. As you can see, it's way thicker than it is in the arteries, and it is almost as thick as the tunica media, or sometimes even thicker in some of the veins. And this contains loose connective tissue, which is of collagen type 1, and it also sometimes contains strips of smooth muscle cells. Now, to summarize everything I just said, and let's talk about the differences between the arteries and the veins. This structure over here is going to be a vein, and this one is an artery. So, the vein at it has a pretty irregular lumen, as you can see, and the artery has a pretty regular round-shaped lumen. And the tunica media is pretty thin, and it gives the vein an irregular shape, whereas the tunica media of the artery is almost like 5 times as thick as the vein, and it ensures that the artery has a round shape of a lumen. And this part consists of smooth muscle cells and elastic laminae, whereas in the vein it just consisted of strips of smooth muscle cells here and there, along with collagen type 3, those connective tissue. Now, tunica adventitia is this outermost layer in the vein you can see, and even in the artery, and it's pretty thick in the vein, and it's almost as the same thickness as the tunica media, or sometimes even thicker. Uh, and also in the vein, it consists of smooth muscles sometimes. But in the artery, it's like pretty irrelevant, honestly. It only consists of loose connective tissue and is pretty thin. Now, let's talk about the arterioles and the venules. The arterioles are basically really, really, really small arteries, and the venules are pretty much really small veins. So these two also consist of the same layers, you can say, tunica intima, tunica media, and tunica adventitia. So the tunica intima is very thin. You pretty much can't even see it in like both of these because you can't really see the endothelium or whatever. It's pretty thin. The tunica media in the arteriole consists of like maybe like 1 to 3 layers of smooth muscle cells maximum. As you can see, like this part is going to be your tunica media, and the tunica adventitia is pretty inconspicuous. In the venules, there's going to be pretty much only one layer, which is also pretty discontinuous, of smooth muscle cells. And you can just see it like this is pretty much all of the venule. So when you compare the arteriole and the venule, you have to look at the thickness for the tunica media. This one is pretty prominent compared to this one. So yeah, that's about it for my part. And now Krish will explain the rest. Before he begins, do any of you have any questions? Okay, Krish, you can start.
Okay, so I'll be talking about precapillaries and postcapillaries in my slides. So precapillaries and postcapillaries are basically a part of the capillary bed, which is basically responsible for microcirculation. Microcirculation again is responsible for transport between blood and tissue. So ion exchange, oxygen exchange, removal of waste products like carbon dioxide, all of that is done using precapillaries and postcapillary venules. So the precapillaries, they also have three layers: tunica intima, media, and adventitia. You might be confused with so many tunica intimas, medias, adventitias. So in your lectures, there's this table which summarizes everything. You could also ask ChatGPT, or you could also search it up to find a whole table, or you could make it yourself to summarize everything in one place. But in precapillaries, you'll have these three layers. The tunica intima, again, loose connective tissue, but very thin. Tunica media, again, smooth muscle cell, but single layer and discontinuous. Adventitia, loose connective tissue, and discontinuous again. The precapillaries have these sphincter-like things. These sphincters are basically formed of like smooth muscle cells. They are going to like encircle the wall of the precapillary. They will help like contract the capillary and control the blood flow. Also help like release local factors to the tissues, which will help in nourishment of the tissues. They also work alongside atriovenous and atrioatriolar anastomosis. These are a few facts of the capillary system which you don't really need to know, but it's just for your knowledge. Postcapillary venules, they are more similar to capillaries, not pre-capillary venules. They're more similar to capillaries because they have continuous endothelium, basement membrane, pericytes. I'll be talking more about pericytes in my next slide. These venules basically have a wider lumen, much bigger lumen than the precapillary venule. Since they have a bigger lumen, they're going to have more connective tissue in their tunica adventitia, more smooth muscle cells in the tunica media, and what this does is then helps the blood regulate the blood flow even better than the precapillary venules. The endothelium is connected with intercellular junctions. What are intercellular junctions? You must know them from the general histology, junctions between cells. So, but in this case, the endothelium has very minimal intercellular junctions. So what happens is, since they're minimal, the cells are going to overlap instead of doing this. This is going to overlap, and once they overlap, they're going to be permeable. And since they're permeable, they're going to facilitate exchange of metabolites, ions, help in water resorption between blood and tissue, and they also help in diaperesis, like Varoon was talking about. They help they help in white blood cells transporting to regions where immune response is required. During inflammation or allergies, where a large amount of histamine is released, venules will allow more of the blood plasma to exit from the tissue. And once that happens, it leads to edema or fluid buildup, which I'll be talking about next. So there's this sort of equilibrium which is maintained in connective tissue, atrial venules, and atriolar ends. So what happens is there is high hydrostatic pressure. What is hydrostatic pressure? It is basically the pressure exerted by a fluid. And fluid in this case is blood, since we're talking about capillaries. So at the capillaries atrial end, water and small molecules like amino acids, glucose, fatty acids, they're going to move into the connective tissue. Over here, you could see the arrows. They're going to move inside the connective tissue and stay there. Once they stay there, and since the water is exiting these capillaries, right, the osmotic pressure is going to increase in these capillaries. And since osmotic pressure increases, the plasma concentration or the plasma protein concentration will increase. The plasma proteins will then pull the water back in. So the atriolar end, the water is pulled out, stays in the tissue. The venule is going to pull back the water or the fluid and take it with it. So this sort of helps maintain an equilibrium. Sometimes what happens is, if the tissue is like in the middle over here and it can't go out, it's going to be stuck here, right? It could lead to edemas. So for that, we have lymphatic capillaries, which again help take 10% of the fluid outside and help maintain the balance. Sometimes what happens is, due to thrombosis or some sort of blockage in these capillaries, the fluid is just going to build up over here and cause an edema or a swelling. It could also happen during inflammation or some sort of allergic reaction. Pericytes. So, what are pericytes? These are specialized contractile cells. Why contractile? I'll be telling you in a bit. They're of mesenchymal origin. Mesenchymal origin basically means they come from a mesoderm germ layer, which is one of the three important germ layers of the embryonic stage. You'll learn more about it in embryology. Uh, they're mostly formed around microvessels, so capillaries, postcapillaries, precapillaries, venules, arterioles. So again, the capillary and the pericytes are going to have these like stellate shaped processes, which are going to encircle the vessel wall and help contract it, similar to what I was talking about before. They help in vessel stability, blood flow. They form this sort of like scaffolding, which is like a net-like structure, which basically supports the vessel and helps stabilize it. They also have a stem cell role, which is mostly for migration of endothelial cells and proliferation of endothelial cells, and also helps in angiogenesis, which again I'll be talking about next. There's three types of capillaries: the continuous, the fenestrated, and sinusoidal. The name itself is going to give you a gist of what they contain, right? Continuous endothelium capillaries will have continuous endothelium, but it's bounded by zonulae oculodentis and nexuses, which are basically these tight intercellular junctions which prevent macromolecules from passing through. Not fully, they're selectively permeable, so they will allow few macromolecules to pass through. They have a continuous basal lamina. Fenestrated endothelium capillaries, however, have fenestrations in the endothelium. They still have the zonula oculodentis and nexuses for similar transfer of macromolecules, but they're going to have fenestrations in the endothelium, which provides a more uncontrolled transfer of macromolecules compared to the continuous type of capillary. Sinusoidal capillaries, they are a bit similar to the fenestrated ones, but they have more fenestrations, they have more holes, they allow more blood to pass through, they allow more macromolecules to pass through. It's not regulated and controlled compared to continuous capillaries. They're mostly present in spleen and bone marrow because blood needs to be transported there, right? So, yeah. Just a gist of what I was talking about: continuous capillaries, continuous endothelial lining, most common type of capillary. They are present in muscle tissue, lungs, brains, testes. The two types of cells present here are endothelial cells and pericytes. The endothelial cells basically help in ECM secretion. So the ECM secretion means collagen, laminin, fibronectin. They also help in vasoregulation, which is basically a vessel. They help in the contraction or dilation of a vessel. They also help in immune responses, clotting factors, hormone or growth factor synthesis. They have a bunch of function. Pericytes, I already mentioned, structural support for the vessel and regulate blood flow by contracting the vessel and helping the blood flow more regularly. This is a picture of the myocardium. You could see the capillaries over here and the endothelium present intermingled with the cardiomyocytes. And again, the summary of everything: the fenestrated capillaries, fenestrations, basal lamina is continuous, found in kidney glomeruli, endocrine glands, intestines. Sinusoidal are mostly, again, they have large gaps between endothelial cells. Basal lamina is absent, which allows free mixing of blood plasma and tissue fluids. This is why they're present in spleen and blood marrow, bone marrow, and liver. Talking about angiogenesis. Angiogenesis is basically a formation of a vessel or a capillary from a pre-existing vessel or a capillary. So over here in this diagram, you could see this capillary. This is straight tube-like structure. It has basal lamina, right? Let's talk about the continuous capillary, for example. It's going to have a continuous basal lamina. This basal lamina, during budding or sprouting, it's going to degrade. So when once it's degrading, it's going to like bend inwards, and once it's bending, it's going to stay there and form like a triangular structure. And in a normal case, when there's degradation of some structure, it's going to destroy the whole vessel, right? But in this case, why it's forming something new is because the endothelium from the basal like from the main capillary, is going to migrate and come over here and attach to the basal lamina using growth factors. So the growth factors like vascular, endothelium growth factor, angiopoietin 1. These are going to like form the capillary sprout. Once that's formed, the endothelium is going to mature and form an endothelial capillary tube, a whole tube, a new tube, which then recruits pericytes, smooth muscle cells, which again allows stability of the new vessel and contraction of the new vessel. Barrier function of the endothelium. This is basically to prevent macromolecules from the bloodstream to go to the tissues. This is achieved through tight junctions between the cells, which, like I said before, allows selective passage of the macromolecules. Not all macromolecules can pass through. And when I say pass through, I don't mean they pass through the junctions. Since they are tight junctions, they're not going to allow anything to pass through. The permeability is referred for the cells. The cells are going to be permeable. So the macromolecules will pass through the cells, not in the junctions. The locations of the tight junctions could be present between endothelial cells. So, for example, in the brain, lungs, thymus, bone marrow, placenta. In the brain, I can give you an example of astrocytes and pericytes. Astrocytes and pericytes have these like stellate-like structures or processes which ensheath the blood vessel and then form the barriers. Between other epithelial cells, in the cases of fenestrated endothelium, they'll form barriers in testes, the blood testicular barrier, which is mostly formed by the Sertoli cells, the blood biliary barrier, the blood ocular barrier, blood retinal barrier, and these endothelial cells have a very low regeneration rate. They only produce like about 1% per day, but the lifespan is about 100 days. The heart. You must have learned most of this in general histology. So, quick recap: the heart has three layers: epicardium, myocardium, and endocardium. Epicardium is the outermost layer. The most outer layer of the heart would be the pericardium. The space between the pericardium and the epicardium would be the pericardial space, which contains this fluid or a serous fluid, which basically prevents friction of the heart during contraction, which enables like smoother contractions of the heart and smoother blood flow rate, etc. The epicardium has mesothelium. Mesothelium and endothelium both are formed from simple squamous epithelium, like we know. Endothelium will mostly be present where there is blood or there's some sort of capillaries or arteries. So in this case, you could see endocardium has the endothelium. The endocardium will also have Purkinje fibers. It has a wavy surface, whereas the epicardium has a more outer smooth surface. The myocardium has cardiomyocytes, which contain uh, the endomysium, which has LCT, capillaries, vessels, intercalated discs, and yeah, that's all.
All right. Thank you everyone for joining, and we hope to see you next week again.